Yeah, so my talk today will, I will try to not focus too much on details on my research. So I'm a neuroscientist by training, but instead trying to focus on some issues where science and art could perhaps meet, which I believe is the purpose of this workshop. So when I was preparing this, it's different from a regular talk that you would do in a scientific conference, right? So I was thinking, so really, what are the connections between art and science? Of course, there are many, and we heard yesterday in the that film about the man who became a particle. Then in the interview with the soprano singer, she said that probably it's creativity that is the thing that connects science and art, and that's for sure. That's a similarity there. But I'm gonna focus on a different thing. Is there a pointer or something I could use? Yeah, there is no pointer. Okay, yeah. So one interesting idea to me is how things are represented in art and in science. So these are two examples how different representations are. So on the left-hand side here it's a famous publication in my field from Hodgkin and Huxley, dated from 1952, where they describe a model for the propagation of action potentials in the nerve cell. So you don't need to bother about the detail, but the form is quite clear. They write some text, and then there are four main equations that describe the propagation of an action potential in a nerve cell. On the right-hand side, there is a picture from Edward Kienholz's The Beanery, which is, you probably know it. It's almost it's not a one-to-one replica of this bar in Los Angeles where he used to go, but you walk in, you really feel that you are in the bar. It has the sounds from the bar. It even has smells from the bar. It's a very nice representation, of course, of this environment, but it's of course not it's an artistic representation. And one thing that fascinates me is so to appreciate this representation of something in the real world, well, you need to go to school, you need to understand mathematics, and it takes a lot of work. Of course, the same is true for this. People will have different appreciations, but there is something to this way of representing things that is much more direct, right? So that's an interesting difference, and if I, as a scientist, could be able to use representations that would convey the message in a more direct way, that would be amazing. So, scientific process, this is my personal take on that, but a very important aspect is to build a model or a representation of the phenomenon that you want to study, okay? And this model should capture the salient features, and it should be of the right type. It should have the right ontology. So that's here, Hodgkin and Huxley, they made their famous model of a nerve cell or action potential propagation. Edward Kienholz made his famous representation of the Beanery. So far it's similar. And then you want to use the model to explain how properties of the model are interconnected, how they work together to generate some interesting consequences. So that's clearly a difference in how representations are used. So we want to work with this. and Hodgkin and Huxley, they laid the ground for a field called computational neuroscience that is substantial, and these very equations are very important in that field. How much people work with representations used in art, that's, I was about to say that's not how it works, but after the very nice presentation of the bacteria yesterday, eating up representations of art, and I thought, no, man, that's exactly what's happening. You are transforming these artworks into new artworks. So that was, it's clearly different from how it's done in science, but it was fun to see. And then, of course, in science, you will compare the behavior of the model with experiments. So that is, it's of course also a difference from how things are done in the art world, I believe. So the phenomenon that I have been interested in is the brain, and in particular how we use our brains to accomplish various tasks. But as you can see, these are two drawings of the brain. This one is we are looking at the brain. If you would cut my head in the middle, we are looking the brain from the side. This is the nose, this is the neck. You can see it contains different parts, and it's, you have the cerebellum, it's this thing here, you have the cerebrum. And here we are looking at the section that is made through, like this, a horizontal section. So we are looking from above, no, from below actually. And so we are getting another view, and you can see this darker thing that follows around. That's the cerebral cortex. And if you, many people, including myself, believe that the cerebral cortex, that's the, if you look over evolution, that's a structure where humans differ most from other animals. Although the structure, the microstructure is very similar, but it has evolved a lot in humans and in apes. So if we look at a small, let's zoom in on a small part of the cerebral cortex, and I'm also using drawings from Ramón y Cajal, Spanish neuroanatomist, and I would say, I would not agree with the speaker yesterday. I would say he is an artist. His drawings are absolutely amazing. You said almost artist or something, but who am I to judge? But I think we both agree. It's and there are a lot of them are available online from the Cervantes Center. So it's really nice to just look at. So this is a section through the cerebral cortex, and all these dots are cells. And what Ramón y Cajal did here is to indicate a few of the cells and to show really how they look. This is not an enlargement of that, but it's another representation. You can see there are different cell types and see how they are connected with each other through these ramifications. So it's a very complex organization, and even today, 100 years later, we still don't know how these cells connect. In a few cases we know, but typically it's still unknown. It's not random, that's for sure. But people are working on the rules of that. But of course, this is the structure of the brain. We are interested in the activity. So the brain is not just a complicated structure. It of course generates activity, and it has activity over many different scales or levels of observation, so both in space and time. So this on the left here, that's a figure from a famous paper by Neer and Sackmann from 1976, where they were the first to record from. So you think of these cells. Now we put a small glass pipette onto the surface of the cell, and this is really it's 10 micrometers square, so it's super tiny. And they were able to isolate one single hole in this cell membrane. And they recorded conductance that goes, so electricity that goes through this hole, the channel, in one single channel. And you can see the time scale here. It's 250 milliseconds. So these reflections here, so it's the channel starts closed, it opens, it closes, it opens. It looks pretty random, but you can appreciate the very brief time scale of these events. So it opens and closes in the order of maybe 5-10 milliseconds. Okay? On the other hand, we can measure the activity at the scale of the whole brain at the same time, using completely different technique. And here, just if we look at, I can't point there, so I will point here. You see there are some small red dots here. So they are in the order of centimeters square. So we are 4 orders of magnitude away here in the spatial scale. And this is what the activity looked like. So this is in response to a visual stimulation. So look at the time scale here. It's 10 seconds. Here was 10 milliseconds. So we again have a 3 order of magnitude different in the time scale. And of course, all the levels in between also exist. I just didn't fit them in the slide here. So we have a structurally complex and heterogeneous system with many levels of activity, from the very tiny to the whole brain, and from microseconds to years. Of course, our brain will change over our whole life course. So what do you do? How do you study such a system? The most obvious thing to do is to give up and study a simpler system, okay? So that was, when I went to grad school, then people were indeed studying simpler system. But if you are young and ambitious and someone tells you want to study the human brain or you want to study the nervous system of a worm, then the choice for me was easy. I opted for the human brain. But in retrospect, maybe I should have gone for the worm, but that you can really understand. So what you can do, I hope, is to focus on a particular aspect and draw on existing frameworks from other disciplines. So one thing that gives a little hope here is that our behavior is not nearly as complex as the activity of the brain. So the number of degrees of freedom of brain activity is infinite for practical purposes, but our behavior is not one-dimensional. It can be, but usually it's a little bit more. But it's not. We are very able to interpret each other and to communicate. So that's possible. So there must be something that makes this complex activity to give it some structure. And people have thought about these things in terms of self-organization. Synergy is Hermann Haken, is a German laser physicist that made this concept very popular. So it's somehow systems that can organize themselves, and I decided to call it coordination. So according to the Oxford English Dictionary, coordination is a harmonious combination of agents or functions towards the production of a result. That's exactly what we want, right? So the activities in the different units at the different levels of the brain must be coordinated with each other and with the environment. And the good thing with coordination is that it has been studied in many different disciplines. So for example, in human movement science, people study how humans move in coordinated ways. So even when I move my arms, there are many degrees of freedom. So each joint will have a number of degrees of freedom, but it's if you think about it and observe, we see that these movements are not independent. So there is some type of coordination going on. Is there a power? Can I? My computer is running out of power.
Yeah. Well, there's.
see, there is no funding in science, so we have very old laptops. We are not allowed to use Apple computers and. Thank you.
That was another choice you should have done.
Yeah, I realized that yesterday. Artists seem to have most fun. Okay, so here's an example, an amazing example of coordination. No. Oh, there is no sound. Huh? Oh, that's. Are you plugged in for sound? Are you plugged in for sound? Yeah. Totally. I'm gonna play it again from the. Thank you. Okay, so this is a favorite example of coordination. This is the musician Dave Lombardo. He used to be the drummer in a band called Slayer, and here's performing War Ensemble live. So just look at how he's able to coordinate his four limbs. Sound. Yeah, that was the. Of course, if you are doing music, then you have cues from the sounds you produce that will help you coordinate. But still, it's a pretty amazing example of coordination. And I was thinking about the gymming talk yesterday when I'm very fond of metal music, and to play drums in a metal band, that must be very, very much like doing workout with this thing, because it's very physical, obviously. And you are definitely doing music. And having fun, yeah. So another example that relates to, it's actually similar to the example that Markus Löffler gave yesterday. These are coordination between different organisms. So this is sardines, fish, and every year there is a very large number of fish that swim up the coast of South Africa. It's called a sardine run. And so they stay together in schools to make it more difficult to be eaten by predators. And there are a lot of predators that try to eat these sardines. Just look at how nice they swim together and how they avoid the predators. There are dolphins. You will see a shark in a little while. You see, they are like a cloud, and it's, as Marcus told yesterday, it's based on very simple rules. Look at how they just avoid. He's not eating any one of them. They are like one big animal. Because apparently, when the predators, if they can't target one single fish, they are unable to eat them. Yeah. So, what does this have to do with the brain? What is the evidence for coordination? Well, apart from that our behavior is not so high dimensional, if we look at brain activity, it was realized a long time ago that it's synchronized. So synchronization means to occur at the same successive instance of time, to keep time with, to go on at the same rate and exactly together, and so on. So neural activity at many different levels is synchronous at local scale and long range. And this has been postulated to be important for various functions of the brain, such as binding together visual features. And this was work from Wolf Singer's group, and that paper there was first published results. So there have been a large number of studies that document synchronous activity. I'm looking at the screen because my screen here is blank. It's another. That's why I'm not facing you. So this is just to give you an impression. This is work I did together with Veronica Nasher, working in Mexico. So there are electric activity from the brain recorded from 33 channels. This is time. So each trace here shows the electric activity. So note two things. It's often fairly, if you look at this activity here, it's kind of repetitive, it's oscillatory. So it's, but that depends on where you are in time, right? And another important feature, which is the synchronization, is that you see that it looks similar in many different channels. And here you see that it says from where these signals are recorded. So there's the top 7 traces are from the right brain, and the others are from the left brain. And you can see that there is long-range synchrony. If you look here, you see that there are synchronous patterns occurring, so in the 2 hemispheres of the brain. This is another example. It's work I did together with David Rob, who is now in Marseille. And this is, the red trace David put an accelerometer on the head of a rat and was recording while the rat was running. And then he noticed that the rhythm, so even when humans are walking, we oscillate slightly up and down. In rats, this is more pronounced, and it's very oscillatory and rhythmic. And he noted that the same rhythm appears in the hippocampus, which is a very important region for navigation, a brain region used in navigation by rats. So how do we study synchronization? Well, as I said before, we need a model. So we should build a model of the phenomenon. It should be a simple model with a periodic motion that has this oscillation. And the simplest generic model is called a limit cycle oscillator. And mathematically, the simplest form you can give it is this differential equation. How am I doing in time? Yeah? It's 5-10 minutes. Okay, good. So it describes the motion of a constant velocity on a circle. So I have a demo that I hope will work. Oh, look at that. So you see there is a small blue dot here. So that's my oscillator, the state of my oscillator. So when I run it, it's going to move around the circle. It's pretty boring. And you see the thing on the left here, it's in grayscale. It represents the position on the vertical of this dot. So when it's down, it's black. When it's up, it's white. This will be more interesting later. We pause. And so hopefully with such a simple system, because it's a generic model, meaning that any self-sustained oscillator should behave similar to this, so hopefully we could capture some interesting dynamics. So what we need to do then is to introduce more oscillators. Oops. So if we now make instead, oh, this is complicated. So now we have 4 oscillators. I'll put them here. Now they are in different colors, and they are slightly different, so they move with different speed. They are not identical. So you can see here the yellow guy just passed the red guy, the blue guy is following here, and they go round and round, and they are not synchronizing. They just follow their own dynamics, okay? Yeah, let me. So what to introduce synchronization. I want to make these oscillators sense each other. So now I have a model, a representation of something that is oscillating. Then I can do that very easily. I can just say that if we have two oscillators, if this one is leading, then he should slow down his speed. And if this one is trailing, he should speed up his speed, so that they would coincide. So then let me introduce a coupling in the model. And we can, of course, have more than 4 oscillators. So now you will see, now there's 100 oscillators, and you will see, so this representation here will show you how synchronous it is in a different way. It will, oh, let me speed it up a little bit. So in the beginning, they are just random. They start at random points, but then due to their interactions, the oscillators will sense each other and they will start to behave coherently after a while, I hope. So you see now it becomes white, it becomes black and white and black. There are still a few ones that are not obeying the synchrony, but they are soon. Yeah, exactly. Yeah, so that's let me close this and get back to. So that's what I did. So this is a very simple model. It's called the Kuramoto model after Yoshiki Kuramoto, who derived it in 1975. And this really is to illustrate the strength of having a representation that you can work with, that you can do calculations with. So Kuramoto was able to solve these couple differential equations exactly, which typically is not possible if you have nonlinear terms, and to show, so in a sense, with such a simple model, we can understand it completely. There are no surprises to be expected. So of course, that shows the strength of having a very exact representation. Yeah, so this is what I'm saying more or less. We can understand such a system exactly, and the representation can be manipulated, so we can work with it. We want to say something about the world, but we represent it in a model, and then we can work on the model and hope that whatever comes out will actually fit our experiments. And if it doesn't fit, then due to that the model is easy to manipulate, we can change it. So this, I believe, it may or may not be similar to when I discussed this with Gustav a lot, and it seems that, I mean, it might be that an artist is thinking a little bit in the same way, that you do one work and then you think, oh, that's good, but I'm not really satisfied, and then you do a new work that builds on the previous work. I suppose that is not so different. The difference here might be that it's very easy to manipulate these representations that we use in science. So me and Gustav Hellberg, it's him sitting there, he's up next. We have been discussing, so how can we illustrate something from using perhaps synchronization or coordination as a principle? How could we turn that into something if we should collaborate in the future in October, first of all? But so I had a very simple idea for how we could perhaps go about do that. And that's to equip people, it's not really clear what type of people, with sensors and explore how they cooperate and synchronize. And that could be done interactively then as a performance. And of course the artistic details I would leave to Gustav, but let me give you, or at least try to give you an example of how this could look like. So here I have an accelerometer that would measure acceleration, and now I hope that it will connect to my computer and display some output. It might not work because there was some problems with, I have to go through the router here and it didn't like, oh yeah. So the idea would be that people would have some type of sensors and move around on the stage and be, oh now it hanged. And depending on, we could use this to produce some more interesting feedbacks obviously than this. This is just the first example. I'm sorry, I don't know why that happened. Kill the plot, yeah. So this is all it's very low tech. It's Arduino, but the fun thing for me is that it's. I mean, I can work with it. I can do it myself. I'm trying to do it again, and let's see. Yeah. So it's a 3-axis. Picks up so acceleration and of course thereby movement in the 3 dimensions. So that's something that we have been exploring, and Gustav will show you some more, or some pictures from when we were fooling around with this. But after having put some work into communicating, so this is the actual accelerometer. They are very small and nice. Then I realized, yeah, of course, and this is probably how we will do it, is that now everybody has cell phones, and these cell phones are, of course, they come equipped with a lot of sensors. So it's just about and then we wouldn't need to put machines on the heads of people. We could just make them download an app to their cell phones, and then we could read the sensor data online. So that would be a much easier way of doing it, but that's something that we realized recently. Yeah, so that's about what I had to say. If you have questions, I will be happy to answer. Thank you.